Yes. So we are going to see the topic molecular diagnostic technique. Okay. Molecular diagnostic technique. Diagnosis the meaning. What is diagnosis? So we have to detect the genetic defect or various other infectious diseases can be detected with the help of various diagnostic methods at the molecular level. So usually the diseases, infectious diseases by bacteria, virus or genetic disease, they show the symptom not in the primary level. So later, final stage only, they will show the symptom or sign outside. So in the diagnostic techniques are very useful at the molecular level. By in this technique, we can use various level of molecular diagnostic techniques. One by one, I'll teach you. So early detection of the disease is not possible. So we are analyzing microscopic technique or blood analysis, okay, or serum analysis, urine analysis. So laboratory techniques, they are not specific also. So we are going to discuss one by one, right? So molecular diagnostic techniques. Now, diagnosis of diseases. Molecular diagnostics na enna? Early diagnosis. Diagnosis na enna? We have to identify the disease before showing its symptom outside. Okay, and we have to identify. We have to find out earlier before in infectious stage is not in the final stage. So early diagnosis of infectious disease or inheritable genetic defects is very important. And only this we can able to give treatment, proper treatment for the disease. So laboratory techniques, I said, they are indirect only. They are not specific, not for particular disease. They are not specific. Scientists are continuously searching for specific, sensitive, simple diagnostic techniques for various diseases. So, mostly there are three important techniques we are going to see in this. One is recombinant DNA technology. Another one is popularly nowadays it is known as Pcr test or polymerase chain reaction, and the third one is enzyme linked immunosorbent assay test. These three techniques, that is recombinant DNA, PCR, ELISA test. So these three techniques help in early diagnosis. You can find out early condition itself, the infectious disease or genetic disease can be found out. Right. So presence of pathogens like bacteria and virus, it is detected only when the pathogen produces symptoms in the patient. So symptoms na adayana, sign. If it shows outside only, we can find out the person is affected by particular disease. The chicken pox, how we can find out? Not after fever, you can see that is small elevation, watery bubbles on the watery surface on the skin surface. It will be denoted as chicken pox. So it shows the symptom like skin rashes. Like this, we can say the symptoms are shown outside. It's not the earliest stage, it is the final stage. But we have to find out the pathogen earlier. By the time the symptoms appear, the concentration of pathogen it becomes very high in the body. So it is very difficult to treat the person completely. So very low concentration of the bacteria or virus, even when the symptoms of the disease does not appear, can be detected by amplification of the nucleic acid. So the bacterial virus, it's very low concentration is there in patient's body. So at the time they are not showing any symptom outside. Yeah, symptom it is showing the external body signs outside by means of eruption, pus, anything. It's shown outside, rashes. That low concentration of pathogens, they do not cause any, do not show any symptoms outside. So, but it can be detected. We can detect it earlier by amplification of the nucleic acid. Whether the pathogen contain DNA or RNA, its small quantity can be amplified. Amplified and multiplied. So that is the technique. So what are the molecular diagnostic techniques I said? So early diagnosis of disease is essential for appropriate treatment. Early detection of disease is not possible. So we are going for early detection of the disease without before showing the symptom, recombinant DNA technology, PCR test, and ELISA test. Even when the symptoms of the disease does not appear, it can be detected by application of the nucleic acid acid. So the first one we are going to see about ELISA test. ELISA test na abbreviation of enzyme linked immunosorbent assay test. Enzyme linked immunosorbent assay test. It is a preliminary early diagnosis of HIV virus disease, AIDS patient we can diagnose. So this ELISA test was discovered by Eva Ingwell and Peter Perlmanin. Peter Perlmanin. It is an important diagnostic tool to determine the person is whether HIV positive or negative in the early stage itself. ELISA test is used both qualitatively and quantitatively, whether they are positive or negative, that ELISA antigen is present or not, its quantity, what is the concentration of the pathogen, low concentration or high concentration, there is quantitatively we can measure whether the virus, HIV virus antigen is present or HIV antibodies are present, their binding is there in the blood sample, we can find out. Depending on the variation, we can use it, it will detect the antigen. Antigen pathogens. These are present in the patient's hormones, enzyme, microbial antigens, and they will bring out what are the drugs we have to elicit, we have to provide, or antibodies. Antibodies are present means we can find out anti-HIV virus, antibodies in the screening test for HIV infection. The person's blood is tested for screening test for this. If it is antibodies for HIV virus is present in the person's blood or body fluid or serum or tissue culture supernatant, then it shows HIV antibodies are there, so the person is HIV positive. Or sometimes we can detect even low concentration of antigen, that is HIV virus, in the hormones and enzyme, that person is HIV positive. Like this, we can find out. So it's discovered by Eva Ingwell and Peter Elmanin. The particular patient is HIV positive or negative. Okay, now we'll see about enzyme-linked immunosorbent assay technique. It is a biochemical procedure. It is discovered by Eva Ingwell and Peter Pellman in 1971. What is their main aim? To detect the presence of specific antibodies in the sample of the blood serum, urine. It is a very important diagnostic tool, measure to determine the person is HIV positive or negative. Now we'll see about the technique. What is the procedure we are using? Okay, now there are 4 important steps: coating, blocking, detection, readout. What are they? Coating, blocking, detection, readout. What is the meaning for coating? Antigen is adsorbed onto the well in the ELISA plate in coating buffer. You can see here the buffer solution and ELISA plate. Okay, sometimes they may have some small depressions. Okay, antigen is adsorbed onto the wells. In the ELISA plate is kept here. If it is adsorbed, na அப்படியே உறிஞ்சு attach ஆயிடும். So into the wells in ELISA plate in the coating buffer. This green color, it is shown inside this, in the middle of in here in the buffer solution, it's called as antigen. Okay, after some times we don't want, we can, that is that particular buffer solution is removed and we can wash the, which plate, the, what is it, ELISA plate can be washed once again. Okay, so this ELISA plate, kept it here, can be washed once again. Remove the buffer once again. If the buffer containing, why we are just washing it? If the buffer containing unrelated protein, we are testing for HIV virus antigen or HIV antibodies. If unrelated, there are the antigens, pathogens, virus, bacteria is there, so there is a chance for these unrelated proteins, they can block the free sites in the cells. So இங்க இருக்கக்கூடிய free sites in the particular region, it can be blocked. To prevent blocking only, the buffer containing, we are just washing it. Okay, buffer containing unrelated protein is used to block and it is free sites in the well. Right. Now, if there is some block in the free sites in the particular region and they can be blocked, that is blocked, and the cavity மாதிரி இருக்குல்லையே அந்த cavity வந்து close பண்ணிடும் with the help of unrelated protein. Once again, this buffer and wash plate. Remove the buffer and wash the plate also. Which plate, ma? This is ELISA plate. Once again, now we are going to detect. Detected enzyme conjugated detection antibody binds to antigen. So this is antigen. Antigen is HIV virus. Now this orange color is, what is it? Antibody or immunoglobulin structure we have studied. So we have added detection of conjugated detection antibody. This is the antibody attached. Okay, conjugate. Here there is an enzyme, this blue color, it is the enzyme, peroxidase enzyme, which can activate this antigen antibody binding. So for the detection of the antigen antibody is this orange color. It is detected with the help of the enzyme peroxidase. Right? Now once again you can remove that buffer solution and wash a ELISA plate. And you can read out, you can see the color change. The substrate is catalyzed by the enzyme to generate color readout. So here colorless solution, buffer solution is changed into yellow color. The color, its intensity, it will vary, it depends upon the amount of antigen present here. The antigen enzyme. So if it is, this is antigen, green color, and this orange color is called antibodies or immunoglobulin. Is directly conjugated, okay, and this is the enzyme directly conjugated to the antibody, and it is conjugated to the secondary antibody and capture antibody. And actually after these two binding, it's helpful for the by the enzyme called peroxidase. We'll see next. During diagnosis, the sample means here we are taking the blood sample containing the antigen is immobilized on the surface of ELISA plate. ELISA plate is having certain cavities and it is immobilized or gets sucked பண்ற மாதிரி உறிஞ்சிக்கிடும் அந்த cavities. After that, the antibody specific to this antigen is added. So first, before that, we are doing the blocking the free sites which are present in the ELISA plate. Then the specific antibodies to the HIV antigen is added. So antigen, antibody. Antigen is immobilized on the ELISA plate. So ELISA plate la kutti kutti cavity irukum adula poi attach aichu. So now it is identified. It is reacting with the antibodies, reacting to the specific antibodies. So what is forming now? Antigen, HIV virus, and the antibodies are added specifically. These two bind to each other, linked to form by an enzyme called peroxidase. So that blue color, I said it's peroxidase enzyme. So if it is unreacted antibodies there. So we added antigen antibody combined conjugated. If it is unreacted antibodies are there, that is also washed away with the substrate of the enzyme hydrogen peroxidase. So here we are adding another one reagent called 4-chloronaphthol. For this antigen antibody conjugation, we are using enzyme hydrogen peroxidase, another reagent called 4-chloronaphthol. The activity of the enzyme peroxidase, it gives that colored product, indicate the presence of antigen. So yellow color change ஆச்சுன்னா HIV positive, presence of antigen. Intensity of the color, எவ்வளோ dark color இருக்கு, it is directly proportional to the amount of antigen. ELISA is highly sensitive test to detect the antigens in the range of nanogram level. Antigen, that is HIV virus, nanogram, minute, nanogram 10 power minus 9 meter, gram here, nanogram. So 10 power minus 9 gram. In that minute quantity itself, if HIV is present in the blood sample, we can detect. So that's why it's called as highly sensitive. There are 4 kinds of ELISA tests: direct ELISA, indirect ELISA, sandwich ELISA, competitive ELISA. Highly sensitive and specific method used for diagnosis. ELISA process the added advantage of not requiring radioisotopes or radiation counting apparatus. In this ELISA test, we are not using any radioisotopes or radiation counting apparatus like this. Only ELISA plate, okay, and antigen, antibodies, enzymes we are adding. Now coming to the second important test, it's called, and it is marked polymerase chain reaction. It is popularly known as PCR test. Okay, PCR test, polymerase chain reaction. It is an in vitro amplification technique. In vitro in the laboratory. Amplification. So what is the virus or bacteria? It's genetic material, RNA or DNA. Minute quantity itself, it can be amplified and used to synthesize multiple identical copies of desired DNA. So it is also useful to synthesize if you are in need of certain desired DNA for a preparation of vaccines, we can produce multiple identical copies of desired DNA with the help of PCR technique. It was first invented by Carey Mullis, the famous person called Carey Bank Mullis. It's his life period is 1944 to 2019. Okay, and this PCR test is now it is Carey Mullis test. It is more popular to test coronavirus nowadays. PCR technique. What are the components of this PCR techniques? We must know desired DNA. We want to get amplification or multiple identical copies of desired DNA means desired DNA, RNA primer, nucleotides, tag DNA polymerase enzyme, and thermal cycler and instrument. DNA sample desired DNA primer to activate or initiate the function various nucleotides. These are the nitrogenous bases, cytosine, guanine, adenine, thymine. Nucleotides, tag DNA polymerase, mix the buffer solution we need. Okay, what is the enzyme used? Tag DNA polymerase. Buffer solution we need, and this is a PCR tube where we are keeping the sample. And everything can be kept in the thermal cycler. So in microbiological studies and all, it's very useful. PCR tests. So we'll discuss about the PCR technique. In vitro amplification technique. In vitro in the laboratory, we are amplifying the desired DNA. Amplification means producing many million copies of desired DNA can also be possible. Many synthesizing multiple identical copies of DNA is also possible. Who discovered this technique? Carey Mullis. And this PCR technique involves 3 steps. One is denaturation, annealing, and extension. What are the 3 steps in PCR test? Denaturation, annealing, and extension. Yeah. Here you can see this is a double stranded DNA. Its polarity is determined by 5' to 3', 3' to 5'. Right? What is denaturation? The double stranded DNA, okay, it is acting as template DNA when it is heated to high temperature, 94 to 95 degrees Celsius. The double-stranded DNA gets separated as 2 single strand. This is called denaturation. என்ன பேரு? Denaturation. Now, annealing, that is the second step. It is PCR test. Here the primers anneal. Annealing na renaturation சொல்லலாம். Annealing na the temperature is reduced, cooling condition, in which the primers anneal are attached to the DNA template. இங்க பார்த்தீங்கன்னா, this is the DNA WLE question. By means of high temperature, they get separated as single strand. Here you can see this is DNA primer and this green color is DNA primer. It can initiate the DNA synthesis and this blue color is called nucleotides. Adenine, guanine, thymine, cytosine. Okay, denaturation printer. The standard template DNA is heated and separated into 2 single strands. Annealing renaturation. The temperature is reduced to 50 degrees Celsius. So what will happen? The primer, okay, here you can see the primer gets attached to the one end of the single standard DNA. So DNA template, this is one DNA template, another DNA template, it is separated here. So here you can see the primers anneal or attach. Annealing attach by means of cooling or reducing the temperature. What is the third step? Extension. It is also called as primer extension. So here once again we are increasing the temperature 50 and 72. When the temperature is raised, so new strand of DNA is made by what is the enzyme used? Tag DNA polymerase enzyme. Here the nucleotide is there. This is the template. Nucleotide is there and this is the primer. Right? DNA primer. It shows its extension. So DNA primer can attach here. This is the 3' of the template DNA, and this is the 5' end of the, what is it called, primer, and it shows its extension. Extend ஆகுதா? அதே மாதிரி here you can see the DNA primer attached to the 3' end. In between nucleotides they form the connection, and this DNA primer, it can show its extension this way. Extension, new strand of DNA is made by reducing the temperature. What is the enzyme needed? Tag DNA polymerase. Now I said the steps involved in PCR technique is denaturation, renaturation or annealing, and primer extension. Okay, so we are using the 3 techniques: denaturation by heating 95 degrees Celsius, annealing or attaching, the DNA primer gets attached to the DNA template in the 3' end, annealing or attaching by means of reducing the temperature to 50 degrees Celsius, and after that attachment of the primer to the DNA template, it shows its extension, and it can produce once again the double stranded DNAs. And this technique, double stranded DNA, first it gets separated, called denaturation, annealing or attachment of the DNA primer, and primer extension. Okay, for one cycle, 3 steps: denaturation, annealing, and extension. So when we are heating the DNA strand, double stranded DNA in 95 degrees Celsius, they separate. Here you can see the separation of the double stranded. Okay, two separate template strands are formed. Then annealing, here you can see this, the primer gets attached, both the side the primer gets attached, and after that this is called annealing. At the time, at the temperature 50 to 55 degrees Celsius, primers bind to the template DNA. This orange color is primer, DNA primer. What is the third one? Extension of the DNA primer. So here, this is a DNA primer. It can extend this side, and this DNA primer, it can extend this side. So both the side, you can see the DNA primers, they can extend and they form double stranded DNA. So from one single stranded DNA, one double stranded DNA by means of 3 techniques, 3 steps: denaturation, annealing, extension, 2 double stranded DNA are formed. Like this, many copies of desired DNA can able to form. So another one important bacterium which is useful, Thermus aquatics. This is a bacterium that lives in the hot springs and hydrothermal vent openings. Tag polymerase, DNA polymerase. It was identified as an enzyme able to withstand protein denaturing condition when it is in high temperature up to 95 degrees Celsius. We said no, they can denature the characteristic feature of the protein. So for that, tag polymerase enzyme can be produced by this Thermus aquatics bacterium. Okay, it is required, this particular enzyme is required during PCR. Therefore, it is replaced by DNA polymerase. So, but I said tag polymerase, it's available only in that particular bacterium living in hot springs and hydrothermal vent, high temperature vent or opening. So, we couldn't able to prepare or produce more quantity. So, we are depending on another same type of enzyme called DNA polymerase from E. coli. So that is we are using in PCR. That original tag polymerase enzyme is not available for us easier. So what we are using? DNA polymerase we are using. So what are the use of this PCR technique? PCR technique oda use enna? It is useful in clinical diagnosis. It is a diagnostic technique, already we have studied. Clinical, for the before treatment we must know the diagnosis of various diseases. So what are the diseases? Diagnosis of inherited disorders, genetic disease, viral disease, bacterial disease can be diagnosed in the earliest stage. Pathogens are present in very low concentration itself, we can find out. And PCR is employed in prenatal diagnosis of inherited disease. Prenatal, before the birth of the baby, delivery of the baby, we can see congenital condition itself. Before the birth itself, we can find out any defect in the baby, and it is called prenatal diagnosis of inherited disease by using chorionic villi sample. Okay, chorionic villi of the from the inner margin of the uterus of the embryo, or cells from the amniocentesis method, we can take and test for PCR whether the baby is normal or it is affected by any inherited diseases. So that's why it's called prenatal diagnostic test. Some other important genetic disorders like sickle cell anemia, beta thalassemia, phenylketonuria, it can be detected by PCR test. And one more, complementary DNA from PCR is a valuable tool for diagnosis and monitoring the retroviral infection. So any kind of retrovirus, mostly they are viruses which are containing RNA. When they are introduced or attack the host cells, their RNA can be converted into, or we can reverse transcribed into, once again into DNA, and after its multiplication, this DNA once again it is transcribed into DNA. So like this RNA conversion to DNA, DNA to RNA, this is called retrovirus. Such retrovirus, the example is HIV, because HIV virus causing AIDS, it is RNA containing virus. When it attacks the human body cells and the human body, that DNA containing cells, this DNA, that is the RNA of the virus, HIV virus, it is reverse transcribed into DNA and they can multiply. Later they can be transcribed into DNA later. One such example for this they are giving, once again, tuberculosis. TB is caused by Mycobacterium tuberculosis. Another one example for retrovirus I said, HIV, causing the disease called AIDS. And cervical cancer caused by papillomavirus can be detected with PCR test and used to detect various sex-linked disorders. Okay, color blindness, okay, hemophilia. In infertilized embryo stage itself, we can find out the various diseases. Okay, sex-linked disorders by means of clinical diagnostic technique. Okay, now we'll see about what are the applications or uses of technique. It is very important in the study of evolution and mainly in phylogenetics. Phylogenetics, evolutionary development, which are useful in the diversification of the species. ஸ்பீஸோட டைவர்சிஃபிகேஷனுக்காக நம்ம எவல்யூஷனில் யூஸ் பண்ணுற வேர்டு தான் பைலோஜெனetics. So for this study of evolution, PCR is very useful. We can amplify even minute quantity of DNA. If we are having very low concentration of DNA from any source, not only from blood, hair, mummified or preserved tissues, bones, or fossilized material, can able to multiply the DNA quantity by means of PCR test. It is used in the field of forensic medicine. Forensic medicine, in the forensic studies, if the criminal's minute quantity of the DNA is available from the sample, we can use this PCR technique and amplify the DNA content and we can match and find out. We have forensic medicine, we can use this PCR technique. It is also useful in this way, it is useful in identification of criminals and forensic medicines for the preparation. Also it's useful and used in the amplification of specific DNA segment to be used in gene therapy. So actually PCR or polymerase chain reaction, it was discovered by Karimalis, used in amplification of whatever specific DNA segment we need. It is very useful for gene therapy also, not only for treatment of various diseases or for identification, diagnostic technique. Apart from this, we can use this also useful for gene therapy. Yes.